right, who remembers anything at all about the spinal cord? Tell me what you know. Reading the slides. Yeah. Okay, so yes, it is a central information pathway from your periphery to your brain. So the spinal cord is part of the central nervous system. It communicates information from the periphery, from your body to your brain. It conducts information up to your brain and then back out to your body. If information is coming into your brain, what type of projections are those called? Afferent. Afferent projections arrive. Afferent arrive. Ascending. Okay, and if they're coming back down to your body from your brain? Efferent. Efferent. Exit. Descending pathways. The spinal cord functions to integrate signals into the central nervous system from multiple sources. So we take in all that information, we integrate it, we make a decision about what to do about it, and then we send an output back out the spinal cord. It's also useful for locomotion and reflexes. We have 31 pairs of spinal nerves that come out of the spinal cord. They pass between the intervertebral foramina, except for the first pair, which comes out just beneath your skull before C1. What are the portions of our spinal cord again? What's this right here? Cervical, and then lumbar. No, right here. Not lumbar. Thoracic, yes. And then down here is lumbar, and then sacral, and then coccyx. Okay. Coccygeal, coccyx. Okay, so cervical, and then thoracic, lumbar, sacral, coccyx. All right. Okay, so we have these nerves coming out all the way down to L1, L2, and then what happens at L1, L2? Sort of. They're not, it's no longer the spinal cord. It becomes the cauda equina. Okay, let's look here closely at the cross section. Okay, so what are the key points that you need to orient yourself when you start looking at a section of spinal cord? What's the very first thing you need to look for? The what? The anterior median fissure. You want to find the anterior median fissure because you know then what orientation you're in, right? So this is the front of the body. Right here. My eyes are terrible. Okay, y'all are going to learn real fast. I can't draw for shit. So if I've drawn something and you're not sure what it is, let me know and I'll try to explain it a little bit. Okay. So anterior median fissure is this big dip because remember fissures are big holes. Posterior median sulcus tells us where we're at as far as the backside of the spinal cord, so whatever is going to be more posterior. So basically, we're in this orientation compared to me. Does that make sense? Okay. What are the other key points here? The ganglia, yeah. So here's your dorsal root ganglion. What comes in through that? Sensory signals. Sensory signals come in to the dorsal root ganglion into, what is this? Dorsal horn. And then they are out through the ventral root. Okay, everybody kind of remembering this? Coming back to you? Yeah? All right. So as we're going down the spinal cord, different areas of the spinal cord have different shapes, different widths, different anatomy. Be able to understand the difference between the cervical, the thoracic, and the lumbar regions just so that you can understand where along the spine you are. The entirety of the spinal cord is enveloped in the same membrane that the brain is enveloped in. It's a continuous membrane. What do we call that? It's a 3-layered membrane. I heard it. Pia is one of the 3 layers. Dura, pia, and arachnoid. What do we call those together? The what? The meninges. Yes. This is the meninges. All the way down. Keep that in mind as we're working. Yes, that's exactly where meningitis comes from. Meningitis is an infection inside the meninges. It sucks. Okay. All right. Reviewing. All these are available for you to review, or I'll post this so that you can review if you want to. There are the meninges. Let's see. Okay. Here are your meninges. Here's your dura, your arachnoid. Here's your subarachnoid space. And here's your pia. You can see that this is a subarachnoid space because it has the spider webby stuff in it. Okay. So we have that dorsal root ganglion, right? And that Wilson said sensory information comes into that dorsal root ganglion. It comes into the posterior horn, posterior gray horn. So remember, in the spinal cord, the white matter is on the outside and the gray matter is on the inside. In the brain, it's reverse, where the gray matter is on the outside and the white matter is on the inside. What's the difference? Yeah, myelination. What does that tell us? Where are the cell bodies? Are cell bodies myelinated? Number 1. Axons are myelinated, right, Lauren? So where are my axons?
White.
In the white matter. Thank you, Juan. All right, so what does that mean my cell bodies are? I heard it. Gray matter. Yeah, so the cell bodies are in our gray matter. Just coming back, because cell bodies are not necessarily going to be myelinated. The axons are myelinated. That's the white matter. It's white because of the myelin wrapped around it. The cell bodies are in the gray matter. So this is clumps of cell bodies in here. So we have a posterior horn. Ooh, I don't know what that is. No, I don't want to get started. Get out of here. You guys, I'm sorry. No. Posterior gray horn, lateral gray horn, and anterior gray horn. And again, orient yourselves so that you can find your posterior sulcus and your anterior fissure. Okay, so we can break these horns down into specific nuclei. So we have sensory and motor. So sensory here at the top, motor at the bottom, somatic and visceral. Visceral is where we're going to go for autonomic nervous system. So you need to know your visceral sensory, your visceral motor. Okay. Y'all remember how cells synapse into the spinal cord? Who wants to talk to me about it? Nobody wants to talk to me about it. How do cells synapse into the spinal cord? We can have it happen a couple of different ways. Okay, so what is a ganglion? A group of cell bodies. Yeah, so we have these dorsal ganglion, or these dorsal root ganglia. So that's a group of cell bodies, right? So there's a cell body here. And if it's coming in here, what kind of information is it bringing? Yeah, in general, sensory information is going to come in. So it's going to synapse onto an interneuron and then send out a motor command, right? So we can have either a 2 or 3 system synapse in the spinal cord. So a neuron can come in and synapse directly onto the motor neuron that's coming out, or it can synapse onto an interneuron. So you can have an intermediate or not. So 2-step or 3-step. Oh no. Okay. Okay, so we have visceral sensory neurons that come in, dorsal root ganglion, come into the dorsal horn. They synapse either into the dorsal horn so that they can go up. Here, this one's going up too. So they can go up to the brain, or they synapse onto an interneuron, which then can then send out a visceral signal through the visceral motor neurons, through the anterior root. So we can have a couple of different configurations here. We have 2 or 3 steps. So information can go either directly sensory, reflexively synapse onto a motor and send out a reflex response. It can synapse onto an interneuron that then sends that information up to the through the central nervous system to the brain, or it can synapse onto an interneuron that then synapses directly onto a third neuron that sends out the motor command. Always in through the posterior side, the dorsal side, and out through the anterior. So this is always sensory. This is always motor. That's why it's important to find your landmarks. Y'all remember your spinal tracts, right? It's okay, we're not even going to do that right now. It's fine. Don't panic, don't panic. We're skipping all of this. Is that it? Are these just not loading? Well, that sucks. My last slide didn't load. That's okay, we can move to ANS. Okay, so are you guys comfortable with your anatomy? You kind of remembering what's going on in the spinal cord? Okay. All right, so the slides are a little different this semester. I've got all of your outlines in here, and I've got study questions for you. I'm not going to go over most of those in class, all right? That's going to be your responsibility to use those to guide you as you're moving through the material on your own, which you will have to do outside of class. Shocking, I know. Study. Okay. So chapter 15 is going to be over the autonomic nervous system. So we want to know the differences between the somatic and the autonomic nervous systems. So we're going to talk about lower motor neurons of both systems. We're going to look at the 2 neuron chains that we just talked about in the spinal cord, and we're going to talk about how the autonomic nervous system is controlled. So let's take a moment, and I want to pause this. Okay, all right, let's start back here. You guys were visceral, right? Autonomic nervous? Okay, tell me your 3 things on autonomics. Nominate a talker. Blanca's a good talker.
Okay, great.
She typically has notes.
Yeah, that's why I nominated.
Autonomic or visceral, it's the internal organs of the body. And then it includes the sympathetic and the parasympathetic. On the sympathetic is the fight and flight, which includes the exercise or excitement or an emergency. So parasympathetic is rest and digest, and it's more related to conserving energy and replenish nutrients. It innervates smooth muscle and glands. And it produces saliva, controlled bladder, and constrict blood vessels.
Perfect. I like it. No bullshit. All right, let's see what y'all have.
We have that.
Some bullshit? Okay. What do you have to add?
Motor system controls involuntary functions mediated by the activity of smooth muscle fibers, cardiac muscle fibers, and glands. The autonomic nervous system works below conscious level to regulate homeostasis, and it consists of 2 physiologically and anatomically distinct, mutually antagonistic components, the sympathetic and parasympathetic parts.
All right. 2 points to Gryffindor for using homeostasis. Thank you, guys. Perfect. Perfect. Good job. Good group work. All right, who's talking over here?
Me.
All right, let's go. This
Okay,
is
so-
somatic.
Somatic, we just have 3 examples that you can apply to real life, which will make it make a little more sense. So one of them is the patellar reflex. So the reflex arc sends the signal to your spinal cord, and then sends the signal to your muscle, and that's when your muscle will, I guess, reflex. And then another example is, say you're walking and then you see an icy patch. So your visual system, when you see it, will send a signal to your brain, which then travels to the muscle, and that will tell you to take action, which is avoiding that path or being more careful, I guess. And then we have another example. So you're swimming in a river or something, and a leech gets stuck to your calf muscle. Your sensory neurons indicate the presence of something that's persistent on your leg, so your skeletal muscles act to alter your position so that that area can be visually inspected.
Oh, so you turn to look at it because it's something that your brain is detecting. Side note, what is it when you have something happening that your brain is filtering out? Are you wearing underwear? What's it called? Habituation. All right, those were great examples, habituation.
Oh,
So this is the opposite of habituation. So this is all functional under our conscious control.
Right.
Somatic nervous system is conscious control of movements, so conscious control of activities. So those are 3 great examples of how you would consciously control your body through the somatic nervous system. All right, who's talking over here? Okay, Lauren, let's go.
So it's part of the peripheral nervous system, and it's responsible for coordinating the body's movements via skeletal muscles. It's responsible for receiving external stimuli. So that's called the reflex arc. And the examples are the blinking reflex, the knee jerk, the gag reflex, and the startle reflex, which a lot of these you'll see in babies more than adults. And then it regulates activities under conscious control.
What's your favorite baby reflex?
When you pick up their hands and they
I like the Moro reflex when they look like baby crawfish.
Oh,
Or they reflexively extend their limbs. They look like a little crawfish. Stick them up in the air. Yeah, and that's all somatic nervous system. It's all conscious control. Even though it's reflexive, it's still conscious control. It's still skeletal muscle, voluntary muscle that's being stimulated. So it's all under somatic nervous system, or SNS. All right, so this is consciously perceived or controlled processes that are processed through the SNS. We detect signals using our senses, and from sensory organs like our skin, those proprioceptor receptors in our skin, or in our ears, and those Merkel cells, the stretch and touch receptors in our skin. Those feed into our central nervous system, telling us how to respond to a leech or an icy patch before you get to that oh shit signal to your brain because you've already stepped on the icy patch and you're about to fall. Completely different signal. Okay, this involves your cerebrum, so we have to go up and consciously think about this. So this is higher order integration of information. Remember, if we've gone up past the thalamus, we are already doing higher order integration of these signals. If you're doing a reflexive movement in response to, what was your first example? The patellar reflex, and the baby reflexes, the Moro reflex, or the Babinski with those are going to stop in the brainstem and the spinal cord. Remember, those are the reflexive centers, so those are the things that basically keep you moving and keep you alive and moving as a human should move. So we stop in the brainstem and the spinal cord with those reflexive movements. If we need to think about it like, oh, there's something on my leg, I need to turn around and look and see what this is, that's going to go up into your brain proper, your cerebrum. Come on, there we go. Okay, so again here, we have sensory information that comes in through the dorsal root ganglion into the dorsal horn of the spinal cord. It synapses onto either an interneuron or a neuron that's going to go up into your brain, into your cerebrum, and eventually a signal is going to be sent out to a motor neuron through the anterior root or the ventral root. And for somatic, or nervous system that's going to innervate a skeletal muscle so that you perform some sort of response to that sensory input. Okay, so for the autonomic or visceral nervous system, this happens below our conscious control. We don't have direct conscious influence over these things that are happening. That's a good thing because a lot of these things help us to maintain what? Homeostasis. Homeostasis is how we stay alive. We're constantly fighting our bodies to maintain homeostasis because our bodies are constantly trying to get out of them. Autonomic nervous system is what helps keep us in homeostasis. So this is where you have smooth muscle innervation. This is where you have cardiac muscle innervation. This is where glands are going to be innervated. It responds to visceral sensory inputs. So sensory inputs from the smooth muscle around your blood vessels. So those baroreceptors that are in your blood vessels that can tell when your blood pressure is changing, they'll send that signal into your spinal cord. It's going to go up into your brain and into your brainstem technically, and you are going to respond to that change in blood pressure so that you can normalize your blood pressure, return to homeostasis, if at all possible. So the organization for the autonomic nervous system is a little bit different. So instead of just having a sensory neuron coming in and a motor neuron going out, we go through an autonomic ganglion. So we have a visceral sensory neuron that will detect a stimulus from one of your visceral organs. So that sensory receptor is going to stimulate this neuron. It's going to come in through the dorsal root ganglion, just like any other sensory information would, into the dorsal horn. It's going to go out through a preganglionic motor neuron. So this is motor neuron number 1, which is then going to synapse into the ganglionic autonomic motor neuron. So this is motor neuron number 2. And that neuron is going to send information back to the tissue that the sensory information came from, so it can execute some response. So it's a 2-neuron system. So the sensory neuron comes straight in, just like every other sensory neuron does. Comes into the dorsal horn of the spinal cord. The ventral horn is going to send out the preganglionic neuron. So neuron number 1 is preganglionic. And the ganglionic motor neuron is motor neuron number 2. So ganglionic motor neuron just means that the cell body originates in the ganglion, not in the spinal cord. Does that make sense? That's why it's so important that y'all remember all of those terms from AMP1. You need to know what a ganglion is. You need to know what a nucleus is. You've got to understand the differences because that's going to help you figure out where these cells are originating and where they're projecting to. All right, so this is a 2-neuron system. ANS is a 2-neuron system. Okay, so for the somatic nervous system, we have a single neuron, a single lower motor neuron. It's extending from the spinal cord, the CNS, to the skeletal muscle. That's what we saw last semester. The cell body originates inside the spinal cord or in the brainstem, depending on where that signal is going to. It's a myelinated axon. It's a large diameter axon. What does that mean about the speed of conduction? It's fast. Yeah. Larger diameter, more myelination, faster conduction of a signal. It releases acetylcholine. What does acetylcholine do when it's released onto a skeletal muscle? Causes it to contract. I heard excites. What were you saying, Caleb? No, you said something. Binds to a ligand-gated channel. Yeah. So acetylcholine, you guys remember this? Acetylcholine is released from the neuron at the neuromuscular junction. It travels across the synapse. It binds to its receptor, which is a ligand-gated channel. Binds to that receptor, causes that channel to open. What happens when that channel opens? Sodium goes in. Yeah, salt in, banana out. Salt and banana. Does that work for you?
Yeah.
Yeah, okay. Okay, so salt in, banana out. Sodium's flowing in. What's that doing to our membrane potential? Depolarizing. It's depolarizing. So we're becoming more positive or more negative? More positive, because sodium is a positively charged ion. So more sodium flowing into the cell. We're depolarizing. What's going to happen when we depolarize at a skeletal muscle? We're going to hit a certain point. Yeah, we're going to hit the threshold that then allows what to happen? Action potential. Action potential. Propagation of that signal down the sarcoplasmic reticulum. So we're going to open more sodium channels. These are voltage-gated sodium channels. They're going to open up, going to have a rush of sodium into the cell, further depolarizing the membrane. That's going to propagate down the sarcoplasmic reticulum, getting to the T-tubules. It's going to change the membrane potential across the T-tubule, which stimulates the opening of voltage-gated calcium channels. What's going to happen then? Calcium's dumped out of the sarcoplasmic reticulum onto the sarcomeres, and it's going to cause a contraction of the muscle. You guys have your notes still, right? Yeah? Review your notes. Okay. See, it's building. It's all making sense now, right? Scary, but making sense. Okay, so when that was somatic. That's what we learned last semester. That's generally the example you think of. So let's look at the autonomic nervous system. Remember, this is a 2 motor neuron chain. You've got one originating in the spinal cord and then one that originates in the autonomic ganglion. Preganglionic and ganglionic. So the first is that preganglionic. This is neuron number 1. The cell body is inside the spinal cord or in the brainstem if you're innervating something up this way. It's a thin projection, it's myelinated, and it projects to the autonomic ganglion. That ganglion, what is a ganglion? Cell bodies. In the central nervous system or the peripheral nervous system? Peripheral, yeah. So the ganglion, you know, is going to be somewhere in the peripheral nervous system. The preganglionic neuron projects to that ganglion, that autonomic ganglion, and synapses onto the cell body in that ganglion. It's going to then activate the ganglionic second neuron. So this is the second neuron in our 2-neuron system. That cell body is in the autonomic ganglion. That's the one that's activated by the preganglionic neuron that started in the spinal cord. So spinal cord all the way down to the autonomic ganglion. We synapse onto second neuron, the ganglionic neuron or postganglionic neuron in the ganglion. The cell body is located in the ganglion. That cell will then be the one that's going to innervate whatever tissue sent in the sensory information. It's a very thin axon. It's unmyelinated. So what does that tell us about the speed of conduction? It's slow. Yeah. It's slower. It's still really fast, y'all, though. It's still really fast. But it's slower than the preganglionic, and it's even slower than the somatic. So somatic, remember, those are large diameter, heavily myelinated. ANS, not so much. So this is going to project to smooth muscle. It's going to project to cardiac muscle, and it's going to project to glands. It's also going to release acetylcholine or norepinephrine. So we talked about acetylcholine activating its receptor, its ligand-gated channel. That was a nicotinic acetylcholine receptor. There's also a muscarinic acetylcholine receptor, and there are a variety of norepinephrine receptors as well. It depends on what type of receptors are expressed in the effector tissue. So the effector tissue has, remember, it sent in some sensory signal, activated the preganglionic fiber in the spinal cord or the brainstem. That's our primary neuron or first neuron. That neuron then comes down to the ganglion, activates the ganglionic or postganglionic cell in the periphery. That thin unmyelinated cell then sends a slower signal back to the same tissue where that sensory information originated. It's going to release either acetylcholine or norepinephrine, and it's going to exert some effect. That effect depends on what type of receptors are present. What does that tell us about the specificity of this? So at the neuromuscular junction, we have nicotinic acetylcholine receptors. If we can have different acetylcholine receptors and different norepinephrine receptors in different ratios in effector tissues, does that give us more or less ability to be specific in our response? More. We have more room to play. We have more room to control things like blood pressure and heart rate and respirations. We want more room to control those things, right? Because those are the things that are keeping us alive. So we want to be specific in our responses. We don't just want to dump acetylcholine out and go contract a muscle. We want to make sure that the response that we're getting is clean. That's why we have more receptors to learn. That's why we have multiple neurotransmitters that can function here. Okay, so here's where your preganglionic cell body originates, in this anterior horn of the spinal cord. Remember, here's your landmark. What is this called? On the screen, I just drew it. What's that? Anterior median fissure. So always find that. So here's your dorsal horn, here's your sensory information coming in. It synapses onto the preganglionic fiber. So here's your preganglionic cell body sending out a projection to it. This is the ganglion. It's going to synapse onto, and again, that's the primary, and here's the secondary. It's going to synapse onto the postganglionic cell, which is going to send out a signal to the tissue that originally sent in the sensory information in the first place. In general. Okay, is everybody clear on this 2-neuron system? Is that making sense? Any questions I can answer on this? Okay. So using this 2-neuron chain allows for neural convergence. This allows several preganglionic motor fibers to synapse onto one ganglion. Does that give us, what does that give us? What does that infer for us? If we have several sources coming in to one ganglion, which is going to send out information to a specific tissue, what does that give us the capacity to do? So there's primary neurons in the 2-neuron chain. We can have multiple primary neurons in the 2-neuron chain coming into the same ganglion before we send a signal to the effector tissue. Faster response, broader response. So we can take in more sensory information. We better understand what our response should be, how we should respond in that tissue, right? We can fine-tune our response by having more or less of these primary neurons, these preganglionic neurons, synapsing onto that ganglion. It's another layer of control. And central nervous system is all about control, even when it means we're projecting to the peripheral nervous system. This also allows for neuronal divergence. This allows branches from one of those preganglionic neurons to synapse with numerous ganglionic neurons. So we can have one of those primary cells coming out of the spinal cord or the brainstem going to multiple ganglia. What does that allow us to do? So for convergence, here's your ganglion, and here's your effector tissue. We had lots of cells coming in and synapsing from lots of areas. This is spinal cord, sorry. This is going to be really bad. I told you guys I can't draw. There, now I'm getting it. Okay, so this is convergence, right? This is lots of signals from the spinal cord coming in to one ganglion. And that gives us extra control, right? So divergence means that from one, make that a little clearer. Here we go. From one preganglionic cell, we can go out to multiple ganglia, which means we're sending to multiple effector tissues. What does that do for us? Why would we need to send signals from one spot in the spinal cord to multiple effector tissues? Broader response, stimulate different areas, change lots of things at once. We need a broad response. We need the body to respond in multiple ways to this one stimulus that we got. We know this sensory input that we got means we've got to change this and this and this and this. When we have a capacity for neuronal divergence, we can change all those things at one time. So this is control. It's just control in a different way. Okay, where are we at on time? Got 20 minutes with y'all? Okay. Okay, so the ANS is regulated by the brain and the spinal cord. So the autonomic nervous system is regulated by the central nervous system. The hypothalamus is the key part of this regulation. What do you guys remember about the hypothalamus? What's my favorite word? Not dehydration senses. No, no. The who, huh? Lizard brain. Yes, it's lizard brain. Yeah, it helps keep us alive. It keeps us in homeostasis. Yes. So all the nuclei in the hypothalamus, remember, those are the ones that are going to keep us in homeostasis. So we have to have the hypothalamus functioning and communicating out from the central nervous system to these autonomic ganglia in order to respond to sensory input to maintain homeostasis. This is essential for activation of the sympathetic and parasympathetic nervous systems. Fight or flight, rest and digest. We'll talk about that in a minute. Those are, your book is going to call them emotional. I don't like the word emotional because everything really is a chemical. Everything we call an emotion is actually just a chemical. It's just chemicals. There's no such thing as love. I'm sorry, y'all. It's all fake.
What?
Love.
Oh.
It's all just chemicals. So the hypothalamus is going to be key in regulating those, what we would call emotional or visceral responses. We say visceral, and this is the visceral nervous system. Those things, those gut feelings, those immediate things that you need to respond to be a human. But the hypothalamus doesn't act alone. It communicates with areas around it. It communicates with the thalamus. It communicates with the pineal gland, the limbic system, and it communicates with the cortex. So what was the limbic system again? Anybody remember? This is your emotional center. Fear, happiness, love. And what about the cortex? What was the cortex for? Use your cortex. Thinking. Who said this? Eddie. Yeah. Higher order integration, higher order processing. Thinking about things in a complex way. So it's beyond our lizard brain. It's beyond those gut responses, those survival responses. It allows us to assess a situation, understand what's happening, and respond in an appropriate way. So typically, if you are afraid of speaking in public, you would flip out if you had to get up here and give a lecture, right? Especially if I told you do it right now, you're completely unprepared and, oh, you haven't seen this information before. But you would assess the situation using your cortex and say, all right, it's okay, I'm not actually gonna die. It's all right, nobody's gonna kill me. I can get up here and I can bullshit something and she'll probably still give me at least a B. That's courtesy of your cortex. Sorry. courtesy of your cortex. So all of those parts of your brain are going to integrate together to make sure that we're responding properly to these stimuli. But remember, these are all of our unconscious responses in the autonomic nervous system. So even though these are things that we're unconsciously changing, the part of our brain that assesses the conscious awareness is still involved.
So what about when you dream? Is that part of it still when you're dreaming?
It can be to an extent. It depends on what you're dreaming about, and really we don't understand a lot about that in dreaming. We really don't know for sure. But there are some aspects to that. I mean, sometimes I used to have this reoccurring dream that I was being chased by knights off of a cliff, like knights in armor and a horse. Yeah. And I would wake up having an asthma attack because I was like breathing so heavily. This was when I was like in second grade. They had like a medieval fair at school, and it just screwed me up for years. So that's a visceral response, waking up because of this breathing in response to something that was not actually even happening. So it's uncontrollable, unconscious. So it can be involved. It's just how, why, when, and how consistently. We don't really know enough about that yet. It's not studied a lot. Yeah, it is. It's kind of cool. Okay, so your brainstem nuclei are going to mediate some of these visceral reflexes, primarily maintenance of blood pressure. That is a big one. The baroreceptors in the endothelial cells lining your blood vessels are going to be in constant communication with your brainstem nuclei to make sure that your blood pressure stays in homeostasis. The spinal cord regulates some of these reflexes. Some of them are sympathetic and some of them are parasympathetic. Defecation and urination are parasympathetic. We'll get into details of that later. Let's wait on that. Okay. So here's your level of here's your level of organization. So we're going to start with the hypothalamus. Remember, this is central nervous system. Here we're integrating things, but remember, the hypothalamus is also taking information from limbic. So here's your limbic system right here. It's your hippocampus. This is your limbic cortex. Here is your neocortex and visual cortex back here, all integrating this information in. The brainstem is going to contain the major reflex centers, so these are going to be things that are basically keeping you alive. And the spinal cord contains a lot of parasympathetic reflex centers as well. Okay, so I'm leaving these slides in. I'm not covering them, but I want y'all to have them. They're available on Canvas, and I want you to be answering these as you're studying, because I'm going to take questions for your exam from these questions on your PowerPoints. You're seeing some version of the test right now. You're welcome. Okay. When you're taking notes on those Google Docs that you're sharing, right, because we're all doing that, right? I would suggest putting in these learning objectives and then typing notes underneath that as you're getting that information from me, or as you're studying and you're getting that information when you're listening to these lectures again. That's going to be a good way to organize your notes. So again, I'm leaving these in. I did minimal editing to these this semester because I can't GIF. I can't GIF in this in OneNote. Makes me, you can't. I know, it's bad. Okay.
I still like the basketball player.
Basketball. He's not it's not a basketball player, Lauren.
I know.
Gosh. Okay. All right, so there are some functional differences between the different divisions of the autonomic nervous system. So the groups in the back both talked about sympathetic and parasympathetic nervous system. So Blanca, you were talking about sympathetic and parasympathetic. Parasympathetic was rest and digest. So these are the things that are going to be, they are energy conserving things. So these are the things that are going to allow you to extract energy through your digestive system. It's going to allow you to relax. These are the restorative things. So sleep is part of parasympathetic. Sympathetic is fight or flight. It's when you really need to push your body to move in a way that is going to potentially save your life, or move in a way that is going to potentially earn you resources to survive. So like running from a saber-toothed tiger, or running toward a woolly mammoth you're hunting. out in the woods here. They're distinct anatomically. So the parasympathetic nervous system is craniosacral. So the preganglionic neurons are in the brainstem and between S2 and S4. So brainstem, sacral, S2, S4. That's where everything from parasympathetic is going to be located. It's all isolated there. The preganglionic axons are long for parasympathetic. The postganglionic axons are short. So that means that the ganglia are very, very close to the organ they're going to innervate. Short axons. The preganglionic axons have very few branches. Does that make sense why?
Because of where they're at.
Because they're long. Yeah, they've got to go a long distance to get to the ganglia. Sympathetic are going to be thoracolumbar, so thoracic and lumbar area of the spine. The preganglionic neurons are going to start in T1 and go all the way down to L2. What happens after L2? What happens anatomically? Cauda equina. Exactly. The cauda equina comes in at L2. So does that make sense why we would stop at L2? Yeah, because it's just a bundle of nerves after that. So the physical difference in the sympathetic neurons is exactly the opposite of what you see for parasympathetic. Here, your preganglionic axons are very short and your postganglionic axons are very long. This means your ganglia are right up against your spinal cord. The preganglionic axons can't have many branches. Does that make sense why?
Because they're shorter.
They're shorter so they can go to multiple ganglia. Yeah. And they're all continuous from your thoracic all the way down to the end of your lumbar, right? So all those ganglia are lined right up against your spinal cord, and you can project from different areas of the spinal column to different ganglia. And you can integrate sensory information over a broader area to have larger responses under a sympathetic nervous system. Does that make sense why we would want a broad response for fight or flight?
Yes.
Why?
Because you have to know.
You have to activate tissues
Yeah.
quickly, right? If you're running from a saber-toothed tiger, you don't want to have to activate your muscles slowly. You want to get those moving. Remember, these are the slowest of the neurons anyway. They're thin, unmyelinated projections. So you want to innervate a lot of tissue at once to get everything recruited, moving forward, getting you away from that saber-toothed tiger. Yeah, you got it. Yes, quick on your feet. Don't die. This is the don't die nervous system. Okay, I like this chart as a comparison. It's color-coded. This is a good study guide. Study me. So y'all can see here where you have the parasympathetic and the brainstem, and down here S2, S4. And we have these pelvic splanchnic nerves. Any idea what those are going to innervate? Uh-huh.
And your bowels.
Urinary and bowels. Genitals and bowels. The brainstem is going to innervate cranial nerves. So y'all remember your cranial nerves?
All 12 of them.
All 12 of them? Review cranial nerves. I'm going to put that as a note here too. Review cranial nerves.
Those are on the inflex too.
Yeah, they are. Everybody going nursing? Yeah, you need to know your cranial nerves for the NCLEX. So review cranial nerves as you're reviewing this so that you can understand why the oculomotor, facial, glossopharyngeal, and vagus are so important. Any guesses? Anybody remember anything about those? I don't want to beat this, but I...
Vegas
Wait, makes you constrict?
No, I meant to say dilate. I don't know what I'm trying to say.
It can
I
do
know
both.
that's
It
something
depends. So
exciting.
the vagus is the one that innervates lots and lots of tissues, all through your body. This is the ass to mouth nerve. Yeah.
Oh, yeah.
Doesn't it shut your body down?
It can. It can shut, yeah, exactly, it can. This is the one if you exercise too much, you start vomiting.
Yeah, that's what I was saying.
Mm-hmm.
That's the one that makes you pass out if you lay on a certain side when you're pregnant.
Yes. That's actually because of blood flow too.
Uh-huh.
All right, so please review those as you're considering parasympathetic versus sympathetic. And remember, this is a good summary of the discussion we've had. So you should know everything in this chart. If you know this chart, then you're solid on the discussion that we've gone over today on sympathetic versus parasympathetic. All in one slide. Okay. Okay, so parasympathetic, this is rest and digest. So the activity is localized. We don't have to innervate a large area like we do with sympathetic, because this is not necessarily a mission critical activity. This is rest. You have long preganglionic axons that influence small areas because they are going all the way down to the ganglion that is right next to the tissue it's about to innervate. Sympathetic activity, we want large areas of activation. So we have very short preganglionic axons. They synapse onto their ganglion and then send out long projections to several effector tissues. This is why when animals are startled, they have multiple responses at once. So I primarily did research on mice and rats. You pick up a mouse and a rat, and they're not used to being handled, they will pee on you, they will try to bite, they will claw, they will run, and they will screech all at the same time. Yeah. Fear. All of that has a visceral component to it. All of that is changes in their blood pressure, it's changes in their brain activity to stimulate cortisol release and to modulate their sympathetic and parasympathetic nervous systems in response just to me touching them. Sympathetic stimulation on the adrenal medulla will be the primary location for hormone release that's going to be associated with this response. In my animals, their adrenal medulla is what's releasing the hormones, releasing the cortisol that's causing them to have this fear response. Sympathetic can engage a single receptor, but it's not common. One example of a single receptor being engaged in the sympathetic nervous system is your pupil dilation. Let's hear your questions again. All right, I want you guys to name all cranial nerves. All cranial nerves, plus the 4, no, specifically the 4 that are associated with parasympathetic coming off the brainstem. And explain the actions of the pelvic splanchnic nerves. This is parasympathetic, so it's urination, defecation, and sex. So again, rest and digest, we're trying to maintain homeostasis under conditions of not being eaten by a predator. Craniosacral and S1, S2. For the craniosacral, we have 2 different types of ganglia. We have terminal ganglia close to the effector cell, and we have intramural ganglia that are actually in the target tissue, in the effector tissue. So you can be very close and send out a short projection and innervate a tissue, or you can actually have a ganglion that's inside the tissue. What time are we at? 38? Yeah, we have till 45. Yeah, it's 38. Okay. Okay. So oculomotor, so cranial nerve 3. Facial nerve, cranial nerve 7, glossopharyngeal, and vagus, which ones? Will be the primary cells you'll need to know for autonomic nervous system. These are all brainstem, right? For oculomotor, we're sending out projections to the ciliary ganglion. The ciliary ganglia is in the orbit, the eye. Those postganglionic cells are going to control the ciliary muscle, so it's going to allow you to focus the lens of your eye and constrict your pupil. So it's going to innervate that sphincter around your iris and it's going to allow for the lens to be focused on objects that are moving towards your face. The facial nerve will extend into the pterygopalatine and submandibular ganglia. These are originating in the pons. Where was the pons? It's the bump on the back of your brainstem. Yeah, as soon as you start to see that widening of the brainstem, that's the pons. So this is going to control the lacrimal glands. What were those? Lacrimal glands? Tears, yes. Tears, so lacrimal glands in your eyes. And the glands of the nose and the mouth. The postganglionic cells from the submandibular glands are going to control submandibular salivary glands and sublingual salivary glands. So the facial nerve is going to innervate tears and snot and saliva. It all makes sense now, right? Tears and snot and saliva.
They put us in the gas chambers of boot camp, and that was all of the things that happened.
Tears, snot, and saliva?
Nope.
Yeah,
Every single one of them at the same time.
that makes sense. Yeah, gas chamber, tears, snot, saliva. Okay, so we're talking about cranial nerve 9 and 10. Glossopharyngeal starts in the medulla, cell body in the medulla, so that's still brainstem, right? And it's going to extend to your ear, and it's synapsing in the otic ganglia. The postganglionic axons are also innervating salivary glands. Parotid salivary glands are going to be innervated by the glossopharyngeal nerve. Vagus nerve, this is the ass-to-mouth nerve. This is the one that has cell bodies starting in the medulla, but it projects extensively throughout the abdomen. So this is going to innervate your lungs, your heart, the digestive secretions. It's going to innervate your liver to change how you store carbohydrates. So activation of the vagus nerve changes your metabolism. It changes your energy reserves. It changes how you can access that energy, how readily you can access that energy. And then it changes how your body physiologically responds to the use of that energy by constricting your bronchioles and your lungs, by slowing your heart rate down. Yeah. It actually can constrict a lot of things. And increasing your digestive secretions. So again, rest and digest. So these are the 4 cranial nerves you need to be aware of for parasympathetic innervation. All right. When you're thinking about these things, so salivary gland is pretty obvious. The lacrimal gland is obvious. Constricting the pupil is obvious, but things like the vagus nerve, we're talking about slowing the heart rate, constricting the bronchioles. Think about the context in which a body would do that. Think about the context in which you would want that to happen. So I can...
If you're in shock, that was what you would want to happen to save your organs.
This is a rest and digest thing.
I wouldn't know what I meant.
You're increasing digestive secretions. If you're in shock, you're not worried about digesting your lunch.
You are resting.
You are resting. You are definitely on the ground. So my concern is that you understand the physiological reasoning behind why these things happen. So I can give you a scenario, and you can tell me based on the things I'm listing out in the scenario if this is going to be sympathetic or parasympathetic. And then based on that, you know where the anatomy is, what's involved, and you could say vagal nerve, activation, cell bodies in the medulla, synapsing onto organs XYZ because this situation is parasympathetic, rest and digest. Okay? Got it? Not in shock. Shock would be fight or flight. Okay, let's stop here, and we'll start with pelvic splanchnic nerves next time.